Good morning. Can everyone hear me? Let me just get a heads up from the back. All right. Good morning, everyone. Thank you for joining Ohio's Urban Forestry Program's 2020 Conference Series. My name is Tyler Stevenson, our Urban Forestry Coordinator. I'd like to welcome you on behalf of all our regional urban foresters. We're excited to bring you day 3 of our month-long webinar series with the latest U.S. Forest Service research straight out of Delaware, Ohio here on ash and elms. We'd like to thank the Forest Service not only for the great lineup of speakers today, but also for their use of the Adobe Connect platform that we're using here to bring our regional urban forestry conferences to your home or office during this time of social distancing. And as always, a special thank you to Donna and Sherry for their behind-the-scenes technical support to keep the wagon wheels rolling. As you'll see, there's a poll question up here at the beginning. I see a lot of you are answering it right now. Dr. Flower had some questions on here. I have this one now. We'll have another one at the break, or another couple at the break, and another couple during their Q&A, just to get a better understanding of the workflow of urban forestry practitioners, get insight into your decision-making process, as well as a way to better direct their science to be more useful. So you'll see some of these questions pop up at the break, like I said, at the break and at the end of the Q&A. So without further ado, I'd like to turn it over to this morning's moderator, our regional urban forester for Southwest Ohio, Wendy Van Buren. Take it away, Wendy.
Thanks, Tyler. Good morning, everyone. It's really great to have you here on day 3 of our conference series. Really appreciate you joining us today and appreciate you sharing your time with us for all these online educational opportunities. Before we get started, we're going to do a couple of housekeeping tips. First off is your audio. Hopefully you've worked out your audio by now. You can adjust it to Adobe, which is in the top left corner, or your own computer, which would probably be in the bottom right corner. The question and answer box is going to be used throughout the program. You can type in your questions there and we'll share them with the presenters at the end of their session. After the question and answers, we're going to take a 15-minute break from 10:15 to 10:30 and begin our second session. These programs are being recorded. Let's see. Oh no. ISA and SAS CEUs will be earned with completion of the session. The certification number you provided at registration will be forwarded at the certification organization with your seat hours. You don't need to do anything to receive your CEUs. Tomorrow afternoon, starting at 1 p.m., we'll be having Roy Montan from Davey Tree Expert Company speaking on creating a culture of safety. And then in the second half, we'll be joined by the great Bill Fountain from University of Kentucky, who'll be sharing the value of benefits of arbor certification and track qualities for municipalities. So be sure to be there tomorrow afternoon. On our 5th day, next Wednesday, September 23rd, in the morning at 9 a.m. again, we're going to have American Forest Sarah Anderson and our very own ODNR Alan Seward. So join us to learn about the factors that we have contributed to the current state of inequitable tree canopy cover in most U.S. cities and how to address the issue. As well as we're going to learn innovative solutions that Ohio communities are using to change how we make decisions and approach our programs today. And the very final Wednesday will be September 30th. We're going to have a short coffee clash to wrap up the conference, make some general announcements we all make at our conferences to keep you all up to date about what's happening here in Ohio. So hopefully you'll join us for that as well. But today, we have some really wonderful folks, a great team of researchers by the USDA Forest Service who are located in the Northern Research Station near Delaware, Ohio. For today's programming, I'd like to introduce 2 speakers. For the first talk this morning, Dr. Kathleen Knight and Dr. Jennifer Koch of the USDA Forest Service will be speaking on managing impacts of emerald ash borer and their progress on resistant trees and insecticide treatments. Dr. Kathleen Knight is a research ecologist. She studies impacts of invasive pests and diseases and provides management options to reduce impacts of these threats to forests and trees. Her research currently focuses on ash and American elm. Dr. Knight earned her PhD in ecology, evolution, and behavior from the University of Minnesota and a BA in biology and music performance at Hiram College. Dr. Jennifer Koch is a research biologist. Her research uses genetics to address forest health issues. She leads a program to breed ash trees with resistance to emerald ash borer, as well as beech trees with resistance to beech bark disease. Dr. Koch is a graduate of Wittenberg University and received both an master's and doctorate in molecular genetics in Ohio State University. So please welcome Kathleen and Jennifer.
Good morning. Thank you so much for having us here today. We're excited to talk to you about our work on impacts of emerald ash borer and our progress on our research on resistant trees and insecticides. Before I begin, I want to briefly acknowledge our many collaborators on this research, and I've got some listed here, of course, on the slide. So unfortunately, many of you have heard of emerald ash borer at this point. This is an introduced beetle that has since spread in North America. The adult beetles emerge from ash trees in the spring through those characteristic D-shaped exit holes, fly off, mate, lay eggs on the bark of ash trees, and then when those eggs hatch, the larva, which is here on the right, burrows down just beneath the bark of those trees and begins feeding. And as it feeds, it creates these tunnels, or what we call feeding galleries. As you get many, many feeding galleries in a tree, as more and more emerald ash borer make those tunnels, it actually disrupts the tree's ability to transport water and nutrients and girdles and kills the tree. And I'll show you specifically how that happens. So this is the basic anatomy of a tree. The outer layer, the bark. Beneath it, you have the phloem, and then beneath that, the cambium. And then in the center there, you've got xylem. And it's the xylem layer, the sapwood part of the xylem layer, that actually transports water and nutrients up the tree. Wow, that animation is not working quite the way it usually does. This is from Joe Boggs, who's been kind enough to share, but you can see the water is going through those layers. Now in diffuse porous trees, many of the outer rings are active at the same time. But in ring porous trees like ash, it's only the very outer layer that's actually active in transporting water and nutrients up through the tree. And that's why it's so important that the damage emerald ash borer does. So it's out in the phloem layer, and then as those larvae get larger, they start to actually score the cambium and that outer layer of sapwood as well. So they're not only disrupting the flow of sugars, they're disrupting the flow of water and nutrients as well. It was first introduced to southeast Michigan accidentally, probably in the mid-1990s. And since then, emerald ash borer has spread to many states and up into adjacent areas of Canada. There are 16 different species of ash in the United States, and all of them are susceptible to emerald ash borer. We have 5 of these species here in Ohio, and I'm briefly going to go over each of those species. First of all, black ash. So each of these species inhabit kind of different types of habitats. They have different characteristics. Black ash is a more northern species, so it reaches the southern end of its range in Ohio. And it typically grows in wet areas, so swamps, riparian areas. Green ash has a very large native range there in green. And it's typically a riparian species. It can, in Ohio, it can be the dominant species along floodplain forests. However, it's got a really big habitat tolerance, so it can be in a lot of different types of ecosystems. White ash, again, a fairly large range. It's found here in Ohio as well as most of the eastern U.S. This is a species that's a more upland species where it typically grows mixed in hardwood forests with other tree species like hickories and oaks and elms. Pumpkin ash is a really cool one. It looks very similar to green ash and is often mistaken for green ash. It typically grows in wet areas and swamps. So in Ohio, we find it co-occurring with black ash and often green ash as well in a mixed stand in a swampy ecosystem. It's got a more southern range than black ash. And then finally, blue ash, which is a really neat one. It has a very small range. It likes to inhabit high calcium soils and can reach pretty high densities in some of those areas. It's got those really cool winged stems, almost like a euonymus. So emerald ash borer is so damaging because it doesn't impact just one species. It impacts an entire suite of species. It impacts the entire genus Fraxinus. So how can we respond to invasive pests like emerald ash borer? And the answer, I would argue, is through integrated pest management. And integrated pest management has a central goal, and this is the goal that I would usually be thinking about for forest pests and pathogens, to achieve both economically and ecologically acceptable pest impacts. And then you use a combination of different strategies, and these strategies are in those circles around the outside, to achieve that goal. So preventative measures, cultural controls, biological controls, chemical controls, monitoring, and mitigating the impact. And I'm briefly going to go through each of these areas just to show the context for the chemical controls that we'll talk about and the resistant trees, which fall under cultural controls. So first, of course, preventative measures, slowing the spread of emerald ash borer. We've heard of, I'm sure, a lot of the, don't move firewood campaign, which is so important. There's also risk management, understanding what type of forests are at risk, how far north emerald ash borer could go. Cultural controls, which include resistance breeding, as well as silvicultural practices in forests. We have not found any silvicultural practices that seem to help with that initial wave of emerald ash borer in forests at this point. So resistant breeding is really the main cultural control that we're focusing on. Biological control, so that's the parasitoid wasps that actually kill emerald ash borer larvae. And so there are some introduced parasitoids that are being used to attack emerald ash borer. There's also native parasitoids like the Leleuthia there on the left that already exist here in North America and are switching over at sometimes opportunistically to attack emerald ash borer. Chemical controls, of course, insecticides injected into individual ash trees to protect those trees. And then monitoring. Monitoring is so important for all of these different kinds of measures because you want to understand the impact of those strategies that you're using and be able to improve them. We also want to monitor emerald ash borer and monitor the host trees, the ash trees in forests, to understand what's happening out there so that we can come up with new strategies. And then finally, mitigating the impact, and this is, of course, very important in our natural ecosystems where we use restoration techniques to plant trees other than ash in places that we want to remain forest that are dominated currently by ash. And then also genetic conservation of ash, so both ex situ conservation, which is a fancy way of saying we collect seeds and we store them somewhere else to keep them preserved for the long term. And then in situ conservation, which is preserving individual trees on the landscape. And so we'll start off today talking about some of the monitoring work, and then I'll move on to talking about the insecticide work and tree resistance. So I've been monitoring effects of emerald ash borer in Ohio forests since 2005. We set up a number of long-term monitoring plots where we've been tracking over 3,000 ash trees individually over time. I have a number of collaborators who have helped with this work. So we started monitoring in 2005, and I'll just call your attention to the legend there. So white circles are no mortality of ash trees, so all the ash are still alive. And then as you go down that list, as you get towards the black diamonds, that is completely dead ash trees in the stand. You'll see that the plots kind of expand over time, and that's not really the spread of EAB, that's the spread of our research funding to be able to monitor the impacts. So in 2005, we started in northwest Ohio, and most of the ash trees there were healthy at that time, although emerald ash borer was known to be in that area. You can see by 2007 there's some pockets of mortality beginning in northwest Ohio. By 2009, we had significant mortality in that Toledo area. By 2011, that mortality, and this is of the ash trees that are 10 centimeters diameter at breast height and larger, so the stem diameter of about like 4 inches or so. So that mortality had spread throughout northwest Ohio by 2011, and then moved into central Ohio as well significantly by 2014, and down into pockets in southwest Ohio. We did get to go back out to these plots and resurvey all of them in 2019, and by that point, as you might have expected, mortality had spread throughout all of the plots. At that point, we had a handful out of that original 3,000 trees. I think we had about 20 trees that were still alive in any of the plots that were that larger size class of trees. We also monitored emerald ash borer population dynamics in a subset of those plots. So we used the purple traps and counted EAB on the traps. So legend is similar. 0 EAB is a white circle. Over 100 EAB per trap is a black diamond. And so in 2008, the emerald ash borer populations in northwest Ohio were pretty high. The emerald ash borer populations in central Ohio were generally pretty low, except for one of our sites. And you can see after that ash dies in northwest Ohio, the emerald ash borer populations crash, but the emerald ash borer populations in central Ohio are building. And then the emerald ash borer populations in central Ohio crash as they kill the ash trees there. So we're going to zoom in on 3 of these forest stands that I monitored: Wildwood up in northwest Ohio, Stratford in central Ohio, and Darby a little southwest of Columbus. Starting in Darby, the green line is the ash trees. So this is the percent of ash trees that were alive. So we're ticking along at 100% alive starting in 2007 when we started monitoring. And we didn't have any mortality until 2014 where we started to get mortality of those ash trees. The emerald ash borer populations are here in the purple triangles. And you can see we caught our first emerald ash borer in 2011 and then started to have a slow increase in emerald ash borer populations. At Stratford Ecological Preserve, we've had 100% ash trees alive when we began monitoring in 2007, and all of those ash trees died by 2011. And you can see the emerald ash borer population rapidly increasing, and then it peaks in 2010 when we have about 50% mortality of the ash trees. That's typical of our stands. That's when we usually hit the peak of emerald ash borer at around 50% mortality. And then the emerald ash borer population crashes, but they never really seem to go to 0. Finally, Wildwood, most of the ash trees alive when we started monitoring, all of the ash trees dead there by 2010, and you see that crash in emerald ash borer populations. And some years we'll get 0 emerald ash borer, but then we often see it flipping back up and catch a few emerald ash borers on a trap. So they're still around persisting in those areas, just at much lower densities. If we look at that same site and divide up those ash trees into healthy ash trees in green and declining ash trees that have dead branches and dieback, we see this pattern. Where we start off with mostly healthy trees. Sorry, my throat's giving me trouble. We start off with mostly healthy trees that are in green. And then in the middle of the infestation, we have a mix of healthy, declining, and dead ash. And then finally, all dead. So after the ash trees die, they break up and they fall down, of course. And this is a floodplain in northwest Ohio. And you can see the piles of woody debris from all of the dead ash that have fallen and been swept along in floods. We monitored the ash trees in the plots as they fell, and we found that they do break up and fall very quickly. Within 2 years after the tree dies, we see over 20% of those trees falling. Within 8 years of mortality, over 80% have fallen. Sorry about that. Fall allergies are really giving me a problem. So there are many seedlings and saplings that remain that are too small to be infested when emerald ash borer first comes through. We do find that the seed bank for ash is very short-lived. After a massed seed year, we'll see seedlings coming out for a couple of years, maybe 2 years. And this is one on the bottom. You can see those long strap-shaped cotyledons, indicating that it's a newly germinated seedling. So implications of our monitoring work for management. Ash mortality and EAB population dynamics do follow a very predictable pattern, and that allows for planning of management actions. We have found that thinning and other silvicultural actions are unlikely to slow mortality during that very first wave of emerald ash borer. And those ash trees do become brittle and fall very quickly and create that pulse of coarse woody debris in the forests. And of course, as many of you know, it's much easier to cut ash trees while they're still alive. Once they're dead, they do become very brittle and dangerous and unpredictable as they're being cut. So planning is really important. We've also seen that native trees can respond rapidly to ash mortality and actually prevent light from increasing in the understory, which is important because you want to inhibit invasive plant species, which may also respond to ash mortality. So I'll now move on and talk about the chemical controls, insecticide work that we've been doing. So there are a number of insecticides that have been approved for treatment of emerald ash borer. I'm not going to go through all of them. In the lower left there you see this wonderful publication that outlines all of the different insecticide options for protecting ash trees from emerald ash borer. This is available online. Dan Herms and Deb McCullough and some others worked together on this. We've actually worked with, and that red arrow should point actually to the imamectin benzoate, or triage. That's what we've used in our experiments to actually look at the question of efficacy, what factors affect how well the insecticide is going to work on a tree. And some of the factors that we've looked at are the initial health of the tree, so the question of when is a tree too far gone to treat, and then the growing environment of the tree. How does that affect how well the insecticide is likely to work? And I'm going to talk about two studies, one in central Ohio and one in northwest Pennsylvania. The central Ohio study was a green ash stand near our lab in Delaware. Charlie Flower led this work. We injected with imamectin benzoate in 2010 and 2012. We had 32 treated trees and 31 untreated trees. And those treated trees started off in different conditions. Emerald ash borer was already there, so some of those trees were healthy and others were declining. So we rated the canopy health of those trees each year on a scale of 1 to 5, where 1 is a healthy tree with a full canopy, a 2 is thinning, a 3 has some dieback, a 4 has over 50% dieback, and a 5 is dead. And those methods of rating canopies are outlined in our Monitoring Ash publication. So first of all, we found the insecticide worked. The insecticide trees are the ones in the filled-in black circles. The control trees are the ones in the white circles. And as you remember, ash canopy condition 1 is healthy and 5 is dead. So those control trees were dead by 2013. And then we just looked at the trees that have been treated and how the initial tree health affects how well the treatment works. And so those trees that started off down there on the bottom as a condition 1 in the black circles, those trees generally remained healthy over time. The condition 2 trees, the white circles there, actually improved in their health and became more similar to those condition 1 trees by 2012 and 2013. The condition 3 trees in those black triangles, some of them declined, some of them stayed the same or improved a little. You can see those large error bars in 2011, 12, and 13, and there's a lot of variation in what happens with a condition 3. But in general, they do seem to decline. Condition 4 in the white triangles typically died. A small number remained a condition 4. So then for the second study, this was an insecticide treatment study that we've been doing at the Allegheny National Forest with a number of collaborators from the A&F, from state and private forestry, and from universities and botanic gardens. This study had 4 different goals. The first was to achieve in situ conservation of ash genetic diversity. We also wanted to test treatment efficacy, test for something called associational protection, and monitor the progression of EAB and ash mortality. So this is the Allegheny National Forest. We set up 27 sites on the forest, a total of 540 trees that were treated. So we treated 20 trees per plot. And on the right here is just an example of what a plot might look like. These are big circular plots. In this particular one, there's 26 dots for 26 trees. 20 of them are treated with the blue circles, and then untreated would be those 6 orange circles. So the plots varied in the number of trees that they had in the plot, but 20 trees were always treated. So we had a question then. So that was the strategy that we chose for treatment of trees. What if we had only treated 10 sites for a total of 200 trees? That would be a lot cheaper, right? Or what if we only treated 4 sites but had 200 trees? So we wanted to understand, if you're going to use this kind of strategy to conserve genetic diversity of ash, how well does it work, and are there better or worse ways to select your plots and arrange your trees to get the biggest bang for your buck? So we sampled ash leaves from a number of trees across the forest, and what we found from some genetic analysis was that the insecticide treatment worked great. It conserved around 97% of the genetic diversity. To get a better bang for your buck, you need to focus on more treatment sites, and doing a genetic analysis beforehand would allow you to choose optimal combinations of sites, which allows you to treat 20% fewer sites if you're choosing good combinations of different sites to really capture genetic diversity. What we found with treatment efficacy, so the Allegheny National Forest has topography, unlike many of the areas I had studied in Ohio. And so we have lower slopes, and on the lower slopes the trees are initially more healthy, and then we have upper slopes where the trees are less healthy. And this graph has printed all of the text in reverse just to keep everybody on their toes here. Okay, So the upper slopes are in light gray and the lower slopes are in black. And you remember that 1 through 5 scale, 1 is healthy and 5 is dead. So on those lower slopes, we see the healthier trees, and that seems to continue over time. So we'll be able to look further at the insecticide treatments in those sites as mortality progresses. We also set up a number of control plots. These are prism plots across the ANF to monitor the progression of emerald ash borer and provide a comparison for the ash treatment sites. So in 2010, most of the ash trees on the forest were green. So in these maps, green is healthy and red is dead. By 2015, we saw some mortality popping up. And that was, of course, even more apparent by 2018 and 2019. We had significant mortality across the forest. And this is also in reverse. The effects of insecticide treatment are really interesting. So the prism plots that are the control plots are in red. And then the yellow and green are trees that are in the treated plots. And the green ones are the ones that were treated with insecticide, and the yellow ones are the ones that were not treated with insecticide. And the y-axis is, again, that ash canopy condition where 1 is healthy and 5 is dead. So you can see those red untreated prism plots going up over time as those trees are dying. You can see the treated trees there in green. The treatment seems to have worked great in terms of keeping those ash trees healthy and, in fact, having some improvement in their health over time. But then what's interesting, too, is those non-treated trees seem to be getting some benefit of being in those treated plots as well. And I apologize for the reversal of all the text on the graphs. I have no idea how that occurred. Management studies of those management implications of those insecticide studies. So the insecticide treatment does seem to work well to protect trees, and it can be used for in-situ conservation by preserving a small number of ash trees in natural areas on the landscape, being able to preserve the genetic diversity of those trees and allow them to continue to breed and reproduce. The treatment works better in trees that are healthy when you begin to treat them. Once a tree is too far gone and emerald ash borer has fed on those trees long enough to really disrupt the flow of water and nutrients in the tree, it's also going to disrupt the tree's ability to transport that insecticide, and so the insecticide does not work as well. And of course, as we're starting to see, the treatment results may vary depending on the tree's environment, so stay tuned for more results on that. Finally, moving on to this piece on tree resistance, we have a number of collaborators on this work as well. So when we were out monitoring the dying and dead trees, we found a single healthy tree at Oak Openings Metro Park. And then we did a complete survey of that area to see if there were any more. What we found was that we estimated there were around 11,000 ash trees in that area that had died. And we found back at that time 284 that were alive, 108 of which had healthy canopies. So that was about 1% of the trees at that site. And that's what we're seeing. At some sites we've got about 1% that are alive, but at some sites we find no trees that are still alive. Almost always the live ash trees were very close to dead ash trees that had died of EAB some time ago. So we have the live ash tree on the left, the dead ash tree on the right. We don't think EAB just missed those trees. We know EAB is really good at finding and killing ash trees. We are continuing to trap, so EAB is still persisting at these sites and may attack those trees. So what's going to happen to those surviving ash trees? We're continuing to monitor them to understand this question. EAB is persisting in those sites. Many of the healthy lingering ash trees continue to stay healthy over time. And this is Rachel Kappler, who's done a lot of great work on this. She's finding that neighboring ash trees may actually impact the health of surviving ash trees. And she's been doing some great modeling work to try to understand long-term dynamics of those trees and management actions that people can take to decrease the probability of extinction of those trees. And of course, we're collaborating with Jennifer Koch to test resistance in these trees and use them in her breeding program. So what is the future of urban ash trees? Should we resign ourselves to their death and use them for poles for basketball hoops? I would not recommend this, as we know they're really brittle. Should we just resign ourselves to having beautiful ash shrubs from all the resprouts of dying and dead ash? These are some in Columbus that die back, and then they resprout from the base. And I think the answer is no, that we can use integrated pest management techniques, a combination of strategies. There's a lot of good science going on, developing new strategies that we'll be able to use to maintain ash and maintain healthy urban forests. And with that, I want to briefly thank the many people Kathleen. Who made this research possible.
Can everyone hear me okay?
And move on to Jennifer.
Okay, I'm just going to proceed. Okay, it's not moving. Do I have control of it?
Those left and right buttons at the bottom left there.
Yeah, I'm clicking them.
Oh, they're not working for you?
They locked up for me, too. I don't know why they're not working for me either right now.
I can always say next slide, please.
Looks like that's the last slide on this.
No.
That is the last
well,
slide there. So we uploaded the wrong one that didn't have Jennifer's slide.
Yeah, let's do that, Jennifer. If you have it on your computer there. While we're waiting for that to load, could you turn up your mic volume a little bit?
Can I turn it?
Oh, never mind. I was going to say, could you turn up your mic volume just a little bit, the down arrow next to your mic? Oh, you can't see it now. Never mind.
Wait, yeah, let me get this started, and then wait, is there a way I can?
Adjust microphone volume. It was back there at the last one you had. Adjust microphone volume. Yep. We can't hear you when you're actually adjusting it. You'll have to hit OK, and then we'll be able to hear you.
Okay, is that any better?
Yes.
Do you want me to turn up higher?
No, that's
Or are we
fine.
good where we're at? Okay.
Yep.
So then everyone can see my screen, and I can just begin?
Yep.
Okay. So I want to thank Kathleen for really setting that up nicely to lead into our research, which is involved with breeding for resistance to the emerald ash borer. And like Kathleen, I need to acknowledge that there are a bunch of people involved in this work. This is not something that we could possibly accomplish by ourselves. I wanted to start by talking about genetic variation in trees, because often I hear from scientists and heard throughout the years that resistance can't possibly exist. The trees have not had the opportunity to co-evolve with emerald ash borer, and this simply is not the case. There are other examples of invasive pathogens and insects that we have found native resistance to. And the reason for this is the tremendous amount of genetic variation that is present in our native tree species. There is actually more genetic variation in trees than there are in human beings. Genetic variation has been the basis for tree improvement programs since the early 1950s. And there are rare alleles or genes that can confer resistance or partial resistance without having that coevolutionary history. They're rare because there has been no selective advantage prior to that invasive threat arriving. So prior to emerald ash borer, it was of no advantage to have genes for resistance to emerald ash borer, so we would expect those trees to be very rare. So I also wanted to talk then about natural selection, which is what is dictating the frequency which with these genes that give the tree an edge over its competition. And in a genetically diverse population, you see a lot of variation in all sorts of traits, growth rates, branching patterns, things like that, even within a species. But you also see variation within the resistance and susceptibility to, say, an invasive insect like emerald ash borer. So when the emerald ash borer comes in, it is going to kill the most susceptible trees, as Kathleen has shown. Not all of the susceptible trees are going to die at the very beginning. It might take some longer than others. But after that peak mortality wave has hit, by definition, there's been a selection event, a massive selection event, and you have increased the proportion of resistant trees relative to the susceptible trees. And that increases the chances that the resistant trees can cross with each other, and that means there's an increased probability that the next generation will have a higher frequency of resistant progeny as well. And that's shown in this bottom row. And you can see there's more resistant trees here, but even when resistant trees breed with each other, some of their progeny will still be susceptible, but natural selection is going to continue and continue to kill those susceptible trees. So after a couple of iterations of this, you're going to have increasing, you're going to be increasing the level and the frequency of resistance within your population. But what happens if the remaining trees are too few and far between, which is pretty much what we're seeing with emerald ash borer? And in this cartoon illustration, there's a resistant tree way over here, and there's one way over here. That's just, a gross oversimplification just to give the idea. And so those two trees would survive, but they're so far apart, they're not going to be able to breed with any other trees. So once they die, even if it's just of old age, that's the end of the line there because they're producing no progeny. So that's where the risk
Jennifer?
of extinction. Yes.
Yes, Jennifer, can you hear me? We've had several people ask to turn your mic volume up a little more. Could you turn it up a few more notches? And for the folks listening in, I had my volume down really low for Kathleen's presentation. I just turned it up for Jennifer's, and it sounded good. But if you're still having trouble, we'll have Jennifer. Yeah, you're in the right spot. Adjust microphone volume. Okay. That one. Yeah, might as well go all the way up.
Okay, how is this, everyone?
Oh, that sounds great.
I'm not seeing anything, so let me know if I need to turn it up higher. So yes, if the remaining trees are too far between and too few, that's when we are at risk of extinction. And we already know from the International Union of Conservation of Nature that 5 of our North American species are considered to be critically at risk of extinction, including green ash. Another reason why I think I have heard for many years skepticism over whether or not resistance really exists is that people don't understand from a genetic standpoint what we're really looking for, what resistance looks like to us. And I found this definition in Nature Reviews, and I've actually now somewhat changed it to make it more specific to trees. But if you read that review, it defines resistance as it's applicable across humans, animals, plants. But the definition of resistance is really the ability of a tree to limit or partially limit insect growth relative to susceptible trees. So what we're talking about is the impact that the tree is actually having on the insect. So if the tree can stop the insect's life cycle at any point, it may buy itself some time. So what we may be looking for with looking for resistant trees are simply those that live longer than susceptible trees. These are not necessarily trees that are immune or bulletproof, if people have heard that expression, but it could be a very subtle expression of partial resistance that can be hard to find if you're not looking very carefully. And the plots that Kathleen and the studies that she has done are really what allowed us to uncover that resistant trees exist. And if they do exist, then there's the possibility that even though they're partially resistant, that they can be improved further through breeding. So what we expect when we're looking at genetically diverse populations is to see a range of phenotypes, a range of types of resistance. And they're going to be a continuum, ranging all the way from highly susceptible, illustrated by this picture on the left-hand side, to partially resistant, to partial resistance, to highly resistant. The susceptible ones are going to be the most common, again, because these trees have not co-evolved with emerald ash borer. But we see, on an infrequent basis, trees with partial resistance. So they're getting attacked, but you can see in this picture that they're able to overcome those attacks and recover. And those that are resistant, where we can't even see a wound on the tree, are incredibly rare. We've only seen 2 of those so far. So we're talking mostly about partial resistance in lingering ash trees. So I do want to make a clarification about lingering ash. We came up with the term lingering ash because we did not want to be overly hopeful until we had data that actually proved that we had resistance. So now lingering ash is really an ash tree that meets these criteria, and meeting these criteria tells us that it's worth further experimentation to determine if it is a tree that has some resistance that would be of value to our breeding program. So the selection criteria, again, based largely on Kathleen's studies, is that we're looking in areas that have reached greater than 95% ash mortality at least 2 years prior, or that also coincides with about 50% ash mortality 4 years prior. The trees have to be large enough to have been infested at peak EAB infestation, which would be greater than about 8 to 10 centimeters. 10 centimeters is what we usually use. In aftermath forests, there may have been time for the trees to actually grow under low EAB pressure, so we may have to do some coring to actually find out what size the tree was at actual peak EAB infestation. And then most important is that we see this healthy canopy. And in this picture, you can see this very healthy canopy, and in the background, there's a series of about 6 dead ash trees right behind it. So once we identify a lingering ash tree, the next step is to go out and collect scion so that we can actually bring that tree and physically bring it into our breeding program. Scion is collected in these pictures using pole pruners. It can be done using a rope saw. Once the scion is collected, typically we start with hot callus grafting because these trees are stressed because they have been attacked by emerald ash borer, even though their canopies remain healthy. They are stressed, so we find that we get better results when we use hot callus grafting. And that's basically when you're just keeping the rootstock and the scion cold and dormant, and you're just heating the grafting to give it a jump start at forming. And that increases our take rates tremendously. Once we have a good, healthy top graft going, we can use that and to take buds off of it and then do bud grafting. And ash is actually a very easy species to bud graft, and you can get a lot of replicates there. So down here are some older trees. Those would be about the age that the following year we would begin testing. So we in a breeding program, one of the next very critical steps is to develop a screen to be able to quantitatively and reproducibly measure resistance. So we had to think about what points in the life cycle of emerald ash borer might the tree be interacting and exert some level of resistance that could affect the life cycle. So we worked with entomologist Therese, and as this slide is depicting, is that there's many different points of interaction, ranging from oviposition to when the larvae is actually growing and feeding throughout the bark layers that Kathleen showed. It's overwintering and then emerging as an adult, where it is again feeding on the foliage. So there may be multiple points that there could be some resistance, and multiple bioassays could be needed to identify the different types of resistance. To have durability of resistance in your population, it's best to have as many different mechanisms as possible. So again, working with Therese, we came up with a couple of different bioassays to measure some of these points of interaction. And what's shown in this slide is a test for adult feeding preferences. So what happens is Therese goes out with her crew and they collect some infested logs. They put them in these collection tubes here. When they emerge, they are attracted to the light and they fly into the cups so they can easily collect them. And then they put a defined number into a cage. And in this cage, there is a series of cut leaves from both lingering ash trees and susceptible controls. The leaves are scanned and measured before and after feeding so that they can quantitate exactly how much foliage has been fed, eaten by EAB on each one. It's a choice experiment, so that there's that aspect of it. The EAB has to actually, it's showing which one it prefers to feed on. And in our first round of studies, 3 of the 8 lingering ash selections that we tested, we identified that adults fed significantly less on the lingering ash relative to the controls. We also worked with Therese to develop an egg bioassay. Therese takes those adults as they emerge. They're able to then group them into mating pairs. They put them into containers in a growth chamber, allow them to mate. At the top of the container, they put a screen, and on the other side of the screen is a coffee filter. That makes the females happy to lay their eggs there, because putting their ovipositor through the screen mimics where they like to lay their eggs in the bark cracks of the ash tree. So when they lay their eggs, it makes it very easy for Therese to just peel off the coffee filters, stick them in a cooler and some Ziploc baggies, and ship them to us. We place them individually on the tree, and that's shown in this middle picture. We do this very carefully because you can see in this picture that even though these are grafted replicates and they've all been treated the same as much as possible, they're different sizes. So we want to make sure that the dose each tree gets is comparable. So we actually measure the bark surface area and space the eggs so that each tree is getting the same dose, or approximately the same dose of egg per bark surface area, so that they can be directly comparable. We do a minimum of 3 grafted replicates of each genotype as a starting point for screening a lingering ash straight out of the forest. The ones that do better will get replicated and tested even farther. 8 weeks after the eggs are put on, they have hatched by then, and in fully susceptible trees, the vast majority of them should have gone through the first 4 instars in their developmental life cycle. But we look at things such as whether or not the egg hatched. We can see the entry hole. We then we mark the entry hole, and then we begin dissecting from the entry hole, and we trace the gallery until we find the actual larvae. If we can get the larvae out without nicking it or cutting it, we get its weight. We record which instar it actually is. And then we also get really happy when we see something like this, which is a larvae that's been killed by the host. So the tree defense responses have killed that larvae. We see some variation in the host-killed larvae phenotypes, but we also see the same type of phenotypes in the known EAB-resistant Asian species, and that's shown in this slide. The upper row is Asian species, the lower is North American species. So the top is illustrating this entry point phenotype, and that's when the larvae it hatches, chews through the egg into the tree, and then dies immediately. So all that's left for us to see is this dark brown circle that the arrow is pointing to. Sometimes we actually even see the back end of the larvae still sticking out of the tree. What's most common is seeing these short galleries that end in a bunch of, there's actual browning of tissue, and we see callus formation around the dead larvae. Other larvae, they don't go in, they stay in the outer bark layers, and you can see there's lignification around their gallery, and eventually they sort of just get, again, encompassed in that response that the tree is having, and they're killed as well. So this is promising that we are seeing the same sort of phenotypes in the known resistant species as we are in the North American species. This slide is just showing you the average. This is the culmination of our first 7 years of work. There's anywhere from 3 to 18 copies of each of these genotypes, and about 3,000 eggs total have been placed on all of these combined. And it's the average per genotype of the number of tree-killed larvae. So, we're seeing anywhere from 0 in some of these, or very close to 0, all the way up to almost 50% of the larvae were killed. We even see like a lot in the 20 to 30% range. And that gives us an indication that even killing maybe 20 to 30% of the larvae is enough to allow those trees to live a little bit longer and explain why those lingering ash are still there. The same with the adult feeding preference. We know that females have to go through a period of maturation feeding before they lay eggs, and they're more likely to lay eggs on trees where they're up in the canopy feeding, because that's where they also then mate. So if they don't prefer that foliage, it's less likely they would lay eggs on that. So it seems that there's multiple mechanisms for resistance. This is just showing the first 4 genotypes that we looked at and marking which ones were significant, either for adult feeding, did bioassay, larval outcome, did the larvae get killed, did it fully mature. We even found significant differences in weight, which could also indicate vigor of the larvae, and it can impact the ability of the adult to lay eggs or the ability for it to even become an adult. So the important information from this is that with the multiple mechanisms, we have the chance through breeding to combine these multiple mechanisms and the genes that are causing them to actually increase resistance within a single tree or within a population of trees. So this is just illustrating that concept that breeding allows combining the best genes from each parent. We know that we don't look exactly like our parents, and our siblings don't look exactly like us. And it's the same concept. So if we have a tree that has the highest larval kill and it's due to a particular gene, we cross it with the one that has maybe the best growth inhibition, the lowest larval weight, and that's due to a couple more genes, and produce many progeny. What we expect is to see these 4 categories in the progeny. You see some of the trees have gotten none of the good genes, so they actually are worse than each of the parents. Some of the trees got the genes from the parents, and so they look like one or the other parents. And then some of the progeny have gotten the best genes from both parents, and they performed even better than either parent. So back when we were able to, we had to kind of wait for the trees to flower in order to be able to do crosses. We do this on grafted, containerized ash trees. So we got a small number of progeny that we were able to test, and that slide is showing the results of that first cross. What these pie charts are showing is the raw data of the number of larvae that entered each tree, all of the ramettes that were tested for each tree. The blue is indicating larvae that made it to the late stages of development. The green is indicating that they only made it to the early stages, early larval instars. The brown is indicating larvae that were killed by a host defense response. And for reference, here's a susceptible control where we saw that all of the larvae actually developed into the later instars, and here is a resistant control of Manchurian ash where all of the larvae were actually killed. So you can see in these 2 parent trees that there is some larvae being killed, but it's a very low percentage, maybe 10–20%. But when you combine the 2, just like the cartoon illustration, we saw some that were worse than the parents. We saw some that killed about the same amount as the parents. And then down here in this bottom row, we found progeny that were able to kill a little bit better, a little bit more than the parents. They had some developmental delays. And then this one on the end is approaching almost the level of what we're seeing in the Asian resistant ash tree. It killed about 3/4 of the larvae that were put on it. So that was a really exciting finding. So since then, this is data from 2018, we've been able to screen more families with different lingering ash parents, and this is showing the results from the 3 largest families that were done in 2018. The arrow is showing the best individual in an unselected susceptible family. So we just used, I think we used a seed lot that was purchased, and we also did a cross with known susceptible trees. And the average of that is at this point. So keep that in mind when you're looking at these 3 families. So again, we're seeing in these larger families that there are trees, progeny that are doing worse than the parents, progeny that are doing better, and progeny that are doing significantly better. So this is showing us that this bioassay does allow us to pick individuals that are showing a great degree of improvement over the original parent trees. So we're up now at over 50%, here at about 65% of the larvae were killed. So our strategy for forest restoration means that we need to be producing seed not only with increased defenses against emerald ash borer and increased frequency within that seed of individuals that are going to be resistant to emerald ash borer, but we also have to retain genetic diversity and the adaptive capacity so that they will not have stress being planted back in the environment that we intend to plant them. And the genetic diversity is critical because these are long-lived plants. They're going to live for a long time. There may be other insect and disease issues that they have to deal with. They're going to deal with issues from changing climate, variations in seasonal drought, things like that. So that genetic diversity means that a portion of at least that population that is produced will be able to survive other stresses as well. So it's really important that we keep genetic diversity when we're talking about forest restoration. So our strategy is to make, with the lingering parents that we have in our breeding population right now, as many different crosses with as many different combinations of parents as we can, and then pick from each of the crosses the best performing progeny and plant them in a second-generation seed orchard. And we anticipate that this seed orchard would produce seed that is now combined, from the best of the best, with even more resistance, a higher level of resistance, and a higher frequency of resistance within that seed. So it would be the first seed that we would consider to be appropriate to potentially start restoration plantings. But I do want to point out that the trees that we're working with are mostly from Ohio and Michigan. So this would not be appropriate for range-wide restoration. Again, we want to make sure we're planting them to places where they're already adapted to. That original stress could impact the resistance. So when we're talking about restoration across the entire range, this process has to be repeated regionally over and over again to produce seed sources that are appropriate for each range. And this is a picture showing the plantation that we have in Delaware of our lingering by lingering families. We have 16 families that have used 14 of the 60 parents we have so far in green ash. 14 of these lingering by lingering families have been screened using our bioassay. We are also planting them out. After the bioassay is performed, we let the stump actually grow back out and then plant them. So they stump sprout for us quite well under the proper conditions. And then we can retest them in the field. So we're including susceptibles right now, but eventually the susceptibles will be removed. So this will be our second generation seed orchard. As we have progressed, I showed data from 2018. Our data from 2019 and 2020 has picked up progeny that have been able to kill 90 to 100% of larvae. Just this year we had 2 trees that had killed 100% of the larvae. These are seedling trials, so that seedling was only tested once, and now it's being tested again in the field. But at the same time, We're going to take those best performing progeny and replicate them so that we can assess them in additional bioassays, so to make sure that holds up through more replication in the bioassays, as well as be able to plant them across various urban environments for potential cultivar development. Some of you might be wondering how much resistance is going to be enough. Of course, that's going to depend on a lot of factors. The hopeful thing is that, of course, the level of resistance that we have is going to be important, and our early data is indicating that at least some of our genotypes are approaching the levels of resistance that we see in Asian ash species. Biocontrol, if it is successful at controlling EAB populations, if it is at least keeping it low and preventing peak outbreaks, or what we saw in the initial wave of EAB, means that we can have lower levels of resistance that are still going to be able to survive. That will prevent the population of green ash from going into extinction, and if those trees can survive just long enough to reproduce. But we also, of course, want that level of resistance that we can use in urban forests and the harsher environments, and we'll be testing for that as well. We know there's an interaction between the EAB population and tree resistance from early field tests of cultivars of both resistant and susceptible tree species, as well as hybrids with intermediate resistance. Those hybrids did really poorly at high EAB. It was black by Manchurian ash, and it died 100% just like the black ash did. But at the lower EAB densities, they performed as well as the Manchurian, or the resistant parent. So we know that's going to have an impact. And of course, we know stress and health is always going to have impacts on the durability of resistance, and that may always be a factor that we'll have to deal with. But that is something that we'll continue to assess as we're looking at the durability of resistance. But what our data is showing so far is that breeding and biocontrol seem to be able to achieve a new equilibrium, or at least our goal is to achieve a new equilibrium under continued natural selection that allows us to have a sustainable green ash resource in North America. So both selection and continued natural selection is going to increase host resistance, at the same time decrease EAB populations through biocontrol to achieve that sustainable green ash resource, and hopefully with resistance that is significantly high enough to be able to withstand the harsher urban environments where we want to put these trees back as well. I want to make, again, point out the tremendous amount of support that we've had, our funding sources from APHIS and the Forest Service, but also some of the states like Michigan and Pennsylvania have actually provided funding for our work. Numerous summer crews and numerous professionals and citizens who reported to us findings of lingering ash. And then really quick, I just wanted to post this for people to take a look at. If you are interested in learning more about identifying and reporting lingering ash, here are some resources for you. We have a website on the Forest Service. Kathleen also showed a publication she had about techniques to monitor for lingering ash. There's the Ecological Research Institute that also has an app. There's the TreeSnap mobile app, or you can just email me with your report. And that was it for me. I think I have to now escape to get back to the regular screen so I can see what's going on with everyone else.
Thank you so much, Jennifer and Kathleen. You guys did a great job. It's really informative. Thank you. Do you, can Kathleen hop on too, and we could do a couple questions before the break? Is that good?
Sure.
Okay, great. So first question is Jason Douglas. He wants to know what the best time to gather ash seeds are and what media to germinate and grow those seedlings.
I see some questions. Are we just reading questions on our own? I can see that someone asked what scion is. That's a good question. I should have specified that. Scion is basically just a dormant branch that can be collected and then through grafting to rootstock, which is just random seedling rootstock, to actually make a genetically identical copy of the tree that the branch was originally taken from.
Great, thank you.
To the question on ash seeds, so I do have a publication on collection techniques for ash seeds. If you shoot me an email, I'd be happy to send that your way. The best time to gather ash seeds is in the fall for all the species, or late summer. It kind of depends on the species and when they mature.
How long
and
before
then
crops
as for
produces
media
flowers? That's another good question. We find that if the flower bud is present on the dormant scion at the point of grafting, it will go ahead and produce flowers right after grafting. So sometimes the year after we graft, we can go ahead and start doing crosses. And then they continue to cross pretty much on the every other year basis that most green ash do.
Hey, Jennifer? Jennifer, can you hear me?
Kathleen, I hope you're looking through some of these questions too. That might be geared for you. Oh, this is another good question. I meant to mention this. Someone asked if I'm looking just at resistance in green ash. I use green ash as an example because that's the program that is the farthest along. We are also working with white ash, black ash, and blue ash. And white ash is starting to catch up with where we're at with green ash, where we're actually starting to do some crosses. Black ash and blue ash were just at the point where we're accessioning some lingering ash of those species, but our early data is showing that the same approach should be successful in those species as well.
Great. Thank you, Jennifer. Kathleen, did you, you'll send us the link for how to grow ash trees. Thank you, and we'll do that one too. Another question, I'm going to do two more, is can insecticide move to surrounding ash trees?
So we're not sure why we're seeing that result with the non-treated trees being healthier. One possibility is that insecticide is moving potentially through root grafts. The other possibility is that because the ash trees are toxic to the emerald ash borer adults that are feeding in the area are killed off, and there's simply less emerald ash borer adults around to lay eggs on those untreated trees. So that's going to be the next line of research to figure out what the mechanism is there.
Great, thank you. I'm not sure what's going on with Jennifer. I'm going to ask one last question, and we'll work out the issue. You'll both stay around towards the end to do a few more questions, right? Great.
Yep.
Rick Harper asked, will ash trees hybridize?
It's possible for white ash and green ash to hybridize. Not super common, but it happens.
Okay, great. All right, well, we're going to go ahead and break for 15 minutes.